So being in a class with only 3 or 4 people is different because there's no eraser. Do you see an eraser in the room? Okay, I don't really need one today, I don't guess. So part of it is it's different because there is no place to hide. so it really is on you guys. In some ways, it's better for your learning because you have to be on every day because there is no place to hide. There's no one to hide behind. Um, the weird thing is that I feel weird just lecturing. So I'm probably going to be trying to engage you more in conversation rather than just trying to deliver material to you because it just seems weird for me to stand up at the board and deliver stuff to the two of you sitting there. So this is the book. When I ask you to read it, because then our conversations can be so much better than if you don't read it, because then I've read it multiple times now, and I come to the conversation, and I have no one to talk to. So that's the first thing, just to kind of get that out of the way. It's a small class, and as a result, it'll have a different dynamic than a larger class will have. Have you guys got onto Moodle yet? Okay. Did you download the syllabus? Okay. Let me pull up my copy. I had to change the office hours because accommodating Emily's schedule to have a time to meet with her, the best time to meet with her was during a time that I had initially planned on having office hours. So I changed that in the syllabus, and I also changed it on the Moodle site. So sometimes I have trouble thinking and doing at the same time. So um, anyway, there's the office hours there. If you need to talk another time, just drop me an email via Outlook setting up an appointment, and I will respond by either saying, yeah, it's okay to meet at that time or not. I use Outlook as my main calendaring thing. So if you send me an appointment request and I respond that I accept it because I'm free at that time, when I accept that, it'll put it on my calendar. You'll be informed that I've accepted it. It'll put it on your calendar. And so, for example, on my calendar for next week is my weekly meeting with Emily. So this is my animal behavior meeting with Emily, where we get together and we go through stuff that we've been through in class together, that she's been watching the videos about. And so that's on my calendar. I know that it's coming. I get a reminder. Generally, I look at my calendar the night before I come to school the next day to look at things that I have scheduled so I don't inadvertently forget something. And so that's the best way to make appointments with me. I made this a hybrid synchronous course just in case we have to go online. My goal is for us to meet in person all semester. That will be facilitated by you staying masked whenever you're in the presence of people in general. I don't know if you guys have been following things. Have you been following things with regards to COVID? Um, just in general, this is the trajectory for Kansas. We are over here. Um, thousands and thousands and thousands of cases. Same thing for Missouri. This is Missouri. Tens of thousands of cases. 26,000 cases on the 18th of January. So be masked in here. I bought a whole pack. I didn't know that the school was going to provide masks, so over the holidays I bought a whole pack of N95 masks that are certified N95 by NIOSH. The ones that the school gave you are not certified by NIOSH, which is a little weird. I plan on being masked until this disaster clears itself up. Um, so I would also encourage you when you're outside of this class, stay masked when you go to the grocery store, et cetera, et cetera. Um, in applied stats, we're going to look at a bunch of data on Friday that show that masking actually significantly reduces the level of spread. So it's a simple precaution, but irritating, but not catastrophic. Back to the syllabus. We will occasionally talk about things that might make you uncomfortable because animal behavior is basically a course about food and sex, feeding and sex. Animals are trying to get food and other resources that they then invest in reproduction. And so sex will come up a lot, feeding will come up a lot. When we talk about competition, we're generally talking about competition for resources, many of which are resources related to gathering more energy to put into reproduction, or in some cases that competition will be for places to build a nest, or in some cases competition over the actual females that a male wants to have sex with. So I'm not going to go out of my way to be insensitive about the topic of sex, but sex is just impossible to avoid. And of course, about animal behavior, a lot of the things that animals do is in the interest of furthering reproduction, and reproduction is the way by which evolution kind of proceeds. And so I may talk about it casually in many cases over the course of this semester. If I offend you, let me know, but it's not out of callousness, it's out of the fact that just in this class, I think about it a lot, so I don't think about it in any way other than it's a means to get to an end, that end being getting your genes into the next generation. So in that way, it can be awkward at times. If it is awkward, let me know, and I will try and alleviate awkwardness as much as possible. And you're probably thinking, probably by just stopping this conversation right here. This is the book. I like this book because of the way it approaches animal behavior. It approaches animal behavior from a oftentimes mathematical and modeling perspective, so trying to quantify behavior as much as possible. At times that requires some fairly complicated math, none of which we are going to do. But thinking about models is useful in helping us think about behavior. And so one of the things that I like about the Davies, Krebs, and West book is that they take that particular approach to it. You will be writing quite a lot in this class. You will write 4 literature summaries. You will write 3 lab reports. So we are going to actually spend a whole lab talking about writing. One of the things you will do for your lab portion of the class is to write a literature summary over a topic that I give you. I probably get those to you in the next day or two so you can get started on it. And a literature review involves reading all the literature that there is to read about a particular topic. So placing limits around that topic are important. That's my job. But then your job is, once I've given you a topic that has limits around it, is for you to research that as deeply as possible to tell us what's known about that particular idea. But then going beyond that, you want to know not only what's known, but you also want to know what's not known in order to figure out what needs to be done to get us to know more about that topic than we currently do. And so we're going to read a paper for lab that does precisely this. It was a paper written in 1990, and it reviews a body of literature, and then it makes a prospectus. It points the way forward for how research in this particular area can advance in the future. And that's the thing that I'm going to be asking you to write for this literature review in And I think of it as a lab assignment. When you write your senior thesis, your introduction is basically a literature review, and your prospectus is at the end of your introduction to your senior thesis, where you say, well, here's what we know, here's what we still don't know, and this is the little chunk of that thing that we don't know that I'm biting off for my senior research. And so, in a way, you should already be preparing for that, because you had to write a literature review for your proposal for your senior thesis. You'll have to have an introduction in your final senior thesis project report, and those are all literature reviews. So knowing how to synthesize literature is an important skill to have as a scientist. So the writing guide that you've been working with your entire program here, you need to have a copy of that. You need to have R in RStudio. We are going to gather data in lab, and we will analyze it, and we'll analyze it using R. If you don't remember R from past semesters, we'll get you reacquainted with it. The lab is not, a lab in R, it's a lab in animal behavior, but we have to do something with the data, and the easiest way to do that is in a language that you're already somewhat familiar with, and Elizabeth will become more familiar with. So if you're having trouble getting those things loaded onto your computers, let me know. If you need access to a computer, because it will not run on an iPad, let me know as well. Be here, mainly so that I don't just hang out in the room by myself. I think you'll learn better if you're here. I'm going to try and avoid doing Zoom. Mason asked me if I could do Zoom today. I've seen people try and do Zoom with people in one place while other people are in a room. We tried to have a meeting that way among a group of faculty, and it was just a disaster because the professor couldn't hear, the person running the meeting couldn't hear the people who were on Zoom. The people who were in the room were listening to the people on Zoom on their computers, and so it was feedbacky, and it's just, I've never seen somebody accomplish it effectively, so I would rather not try. So if we do end up having to go online, we'll probably just be all online or all together. If you have to quarantine, like Mason is having to quarantine right now, I had a couple of emails back and forth with him earlier today. He's just going to look at the videos and then get back to me with anything that he doesn't understand. So that is that. We have a small community. Once again, we will try to be civil, although there's not a lot of hot button topics to talk about in animal behavior. Honor code applies to everything. Do your own independent work. Don't plagiarize, et cetera, et cetera. Do we need to go through plagiarism again? Because I'll do it if we need to, but I won't do it if we don't. You feel like you know what the limitations are. Okay, good. So there was talk in the biology department some time ago that we need to do a thing on plagiarism in each class, and I think we do something in each class in the core. Hopefully by which time you've kind of got the message on that. So if you guys feel comfortable about that, we will forego that. If you have a learning accommodation, you need to let me know as soon as possible. I need to get that from Missy Henry, and then we'll work out whatever those accommodations look like in the context of this class. So we will always wear a freaking mask covering. Ugh. Face covering. Once again, these videos will be available, so anytime you miss class, they will be available within the day of the class being given. Typically, I'd go back to my office right after class and upload them to the YouTube site. The YouTube site will have the link to the playlist on YouTube, so all you have to do is click on that link. It'll take you to the playlist. I don't have it set up yet for this class because I haven't made a video for this class yet. But if you go to YouTube, when I get onto my account, the link will actually take you to a particular playlist. Whoa, sorry, go back. will take you to a particular playlist. So I have playlists from semesters past, but for now there's an applied statistics playlist. If you click on the full playlist, what it does, it just gives you the list of all of the videos that are in there. And assuming that I'm uploading them in a timely fashion, they're listed in date order, so you can find them pretty easily. And I always title the files by the date first, year, month, day, and then it's just a long list. This is from earlier this morning in the data science class that I taught. So they are there. And you can listen to my horrible voice on video. It is frightening. I've also asked you to take a pretest. I sent you an email about that. It is up and running. I can see that, oh, Mason has started working on it. So that's good. Get that done within the next 24 hours, simply so that's out of the way. Like I said before, you get credit for taking it, completing it. It doesn't really matter what your score is. That's not where your grade is coming from. Your grade is coming from completing the task. These are the dates where we won't have class. Assignments are due when they're due, unless there's some extenuating circumstance that you can tell me about. Feel free to contact me via email. You can also just come by my office. Last day to drop is the 18th. So as far as course requirements are concerned, there are 3 exams, 4 literature summaries, 4 literature discussion grades, and 3 lab reports. Oh, and something that's not on there, which is a literature review. Yeah, the literature review is not there, but it's in the list of assignments down below. So when we meet to discuss literature, each time we discuss literature, one of you will be assigned the task of discussing that literature. I'm going to give you those assignments early in the semester so you know which one you're on for. The unfortunate thing about knowing who's on the hot seat for that day is that everybody else in the class thinks that they can just kick back and not do anything, which is not conducive to talking about a piece of literature. So you will, on the day that we're discussing literature, everybody in the class will get a rubric filled out for them about their discussion of the literature that day. And the person who is facilitating discussion, which is one of the 4 of you, or one of the 4 of you, because on the day that Emily is due to be the facilitator, she's actually going to skip her other class that she's taking that occurs at the same time. She's going to be here to be with you guys. So there is a rubric for the facilitator, there's a rubric for the participants, and this is just meant to ensure that everybody has read the article and everybody is participating fully. I'm a participant in the conversation as well, but when you, a student, is facilitating, you're the one who's kind of in charge, and you can treat me just like another student. So that's why there are literature discussion grades in there. That's what that is about. So we have 3 lecture exams, we have a pretest, we have literature summaries that are going to be graded much in the way they were graded in evolution and ecology, with a couple of important distinctions which I'll make note of today. Literature discussion, there's the literature review, which is this long review paper that reviews the literature about a topic and then projects what should be done in the future on that topic. Then we have lab reports. Tomorrow we're going to be working on the Bean Lab. It's a lab that is about bias and how our perceptions aren't necessarily correct in many cases. Then we're going to, once the weather warms up a little bit, we're going to go out and we're going to observe flocks of birds behaving up at Smithville Lake and look at the idea of vigilance, the tendency for birds to keep an eye out for danger, and that tends to be influenced by group size. And so we'll spend a couple of labs gathering data on bird behavior out in the field, and then we'll come in and we'll analyze that, and then you'll write a report about that. And then a big chunk of this lab is going to be a project on aposematism. This grows out of a project that a group of students did in evolution and ecology a number of years ago, where they made clay models of caterpillars and put them out into the field. One model of the caterpillar was a non-toxic, non-aposematically colored caterpillar. The other caterpillar was a monarch model. Put them out in the field and then left them there for a little while and then came back and looked and saw how many birds had attacked the two types of models, which is similar to a paper that we'll actually read looking at this in coral snakes. And so it's a variation on a theme. In order for us to do that, we're going to have to spend some time in lab making the models themselves. And so we'll spend some weeks making models. Right after spring break, we'll put those models out into the field, then we'll pick them up a week later, we'll collect the data off of them, and then we'll analyze that data, and then you'll write a lab report for that. And then there's the final exam, which will be partly just on the last material that we cover, and partly it will be comprehensive. So that is that. There's 800 points in the class. Whatever percentage of points you get is what you get. This class used to be graded using specifications grading. Students hated that because it was different from how they're graded in the past. So rather than continuing to fight that battle, I'm just going back to a more traditional type of grading scheme. What that means is that I'm having to revamp all of my rubrics that I use for evaluating everything so that I can put points on those things rather than making them pass-fail assignments, which is what they were before. So there are going to be rubrics that appear on the Moodle site, but they're not there yet because I haven't finished revamping them yet, redoing them to go back to a more traditional way of grading. Questions about the syllabus? Okay, let's go to Moodle. So at the top of Moodle is stuff that you will find useful. Let me actually rearrange some things here. So for Friday, I'm going to post, actually it's already posted. Let me actually make it visible. For Friday, I want you to read this paper by Joseph and Pinchot in 1989. So we're going to discuss that literature on Friday. I realize that you only have a couple of days to read it. I'm going to facilitate discussion on Friday so that you can see how I want you to be facilitating discussion. So read that between now and Friday. It's not a very complicated paper. It's relatively short, and we're going to introduce the paper today. So read it, understand it. You don't have to write a literature summary about it or anything else. Just come ready to discuss it, and I'll lead discussion that day so you can kind of see what I'm looking for. So it's now visible, and actually that's not the 24th. I have to change things around. That's going to be, what is today? The 19th is 21st, actually. And you will see that what I typically do is I'll generally put a day's material in a folder, and then everything you need is in that folder. And so this is what it looks like when I look at it. I have folders for all of these dates that I just need to change the dates on. When you look at it, what you should see is just the stuff at the top, and then the first week is there, and all the other weeks are blank. The stuff is there, I just haven't made it visible yet because I'm in the process of changing dates on things and also updating PowerPoint. So I went through Friday's PowerPoint and made some changes to it. It's in the Friday material stuff in the form that I want it to be for Friday, but this is what it looks like to you, but most of the stuff is still there. So I put a copy of my weekly schedule. If you need to get a hold of me sometime outside of office hours, these are my weekly commitments that are set in stone. So it has my office hours, and it also has when I have stuff that I can't move. So if you want to meet me at 9 on Thursday morning, I can't do that. That's already marked off for my time with Emily. So that's there. My office hours are on the Moodle site so that you know when those are. The syllabus is there. The daily schedule is there. Let's look at the daily schedule real quick. In the daily schedule, I have, let's see if we can make it bigger so you can actually see it. Basically, what's the topic? What I want you to be reading. So there are 2 papers that are in the materials for Friday. The one I want you to really concentrate on is Joseph and Pinchot. There's also a paper in there by Bateson and Lalande, which I want you to read, but that paper we're not really going to be discussing until Wednesday of next week. I'm sorry. No, sorry. We're going to be covering on Monday, so have that read by Monday because it has to do with the material that is in Chapter 1. So we're going to be talking about Chapter 1 stuff on Monday and Wednesday of next week. Thursday of next week, we're going to have Literature Lab. Tomorrow, we're going to have the Bean Lab. The Bean Lab we should finish, and we should have all the data collected and analyzed by tomorrow. So we might talk about the Bean Lab a little bit next week at the beginning of class, but most of what we're going to be doing next week in lab is talking about this literature review from 1990 that I need to get up on the Moodle site as well. The Bean Lab report is due on the 3rd of February, so you're going to have a short report on the Bean Lab. I'll talk about what needs to be in that report tomorrow in lab. But then on Thursday, once again, assuming that weather is conducive to it, we're going to go out and we're going to observe some bird behavior. We'll do that for a couple of weeks. Then while we're processing the data from the bird vigilance lab, we'll start making caterpillar models in lab. So we'll come to lab for 2 or 3 weeks and we'll just make models, which are time consuming to make. Once those models are made, we will deploy them, like I said, right after spring break. We'll collect them the next week. We'll analyze the data. We'll talk about kind of how that lab report needs to be structured, and then you'll have until the last week of class to get that lab report done and completed. And we won't take up lab time. You can actually repurpose that lab time as time to work on your final report. So oh, sorry, I need to move that. Nope, those last two lab days we won't have lab. I need to move that lab report to the last week, not the week before. So yeah, just 2 weeks at the end, because I want you to have time to do an adequate job. And I don't know what we would do in lab because we've completed a project that you need time to process and think about and write about. And so we could do another activity, but then I don't know what the product would be that would come out of that. So what I've tried to do is give you the topics that we'll be covering, the chapters that they apply to, and then when exams are. And the last couple of times I've taught animal behavior, we've been to stay more or less on topic, on schedule. If we get off schedule slightly, we'll just change what topics are on the exam. We won't change when the exam dates are, so you can kind of bank on those exam dates. So we have literature, Parker and Simmons on the 16th, Brody and Jansen on the 28th. McClintock and Ewitts on April 1st, and then Kroll and Hodel on May 4th. And so those are the literature summaries, and like I said, I'll assign each person who's doing what. By Friday, I'll have that decided. So, the date and time of your final is down at the bottom. I put the applied stats final in there also just as a reminder to myself. That's more for me than it is for you. And that is the schedule. Questions? All right. There's some things on the Moodle site that are meant to help you. So, for example, We're going to be talking about Joseph and Pinchot on Friday. So one of the things I did a number of years ago is I sat down and I wrote what I think would be an A caliber literature summary of Joseph and Pinchot. And so it's there for you to read and look at how I'm structuring it, how I'm thinking about what next steps are, et cetera, et cetera. So that is there for your use. Um, some guidelines for writing papers. Once again, you're going to be writing this literature review, you're going to be writing literature summaries, et cetera, et cetera. This is just a list of things, a description of things that are good for you to think about as you're writing. You would find this useful for any writing that you're doing in this class. You would also find this useful for your senior thesis as well. So once again, I'm not going to necessarily go through this, but it's here for you to use. I've also provided you sample exam questions. If you're wondering, how do I ask questions on animal behavior tests? This is how I ask questions on animal behavior tests. So you can see it's not any different from the way I've asked you questions in other classes that you've taken with me. Um, do, 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 do. So today, if there are no other questions about Moodle or about the syllabus or the schedule, yes, ma'am. Yeah. Well, I think they were. Oh, yeah. Ah, yes. Okay. You are correct. Um, I have shifted somewhat, and you can do this with multiple choice. Well, now I forgot about the test. So in specifications grading, specifications grading is this really interesting idea that basically relates in a very direct way your grade in the class to what you can demonstrate that you can do. So specifications grading has rubrics that you give students beforehand, as we oftentimes give you rubrics beforehand, but by focusing on the tasks that are necessary. So what do you need to do in a literature summary, for example? You need to summarize the article adequately. You need to summarize the article while including enough of the important details that the person can understand what the article was about, not delving so far into the details that the thing becomes too long. And it's just, I might as well just go and read the article myself. The literature summary needs to do that accurately. The literature summary needs to do that with good grammar. The literature summary needs to have a section of critical thought that is forward thinking, that makes the student think about what's still not known about this particular topic and what could be done to delve into these unknowns in the future if one was going to continue pursuing this idea. And so let me just go and get it. So back in the day when I did this other form of grading, I had rubrics, which I've deleted because I decided to change the way I was grading. Give me a sec. So for the bean experiment, for example, The grading rubric would look, oh crud, what happened? I'm no longer there. So this is what the grading rubric used to look like. And so their formatting expectations. Is it doubled space? Yes. These are all easy things. Everybody can double space something. Everybody can type in Times New Roman 12-point font. Everybody can put 1-inch margins. If you can't do these kinds of things at the junior or senior level, then we have failed you in some fundamental way. You should be able to operate Microsoft Word. But in terms of science, did you frame an introduction that leads logically to the hypotheses? Is your introduction factually correct? Are your hypotheses clear, and do they conform to the structural convention of how hypotheses are stated? Are the methods clear? Do they follow a logical order? Are they complete enough that you give them another student, they would be able to replicate the experiment and analyze the data? Results are clear and well organized. So this is for the lab report. So you want to be more in depth than you would in a literature summary. Are the results clear and well organized? Do they include references to figures and statistical results? Are they interpreted correctly? Same thing with figures, discussion, blah, 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 blah. And if you check all these things off, if you've done all these things, you get little checks next to these. And if you get checks in front of all of these, you've passed the assignment. The assignment is pass fail. You know what things are required of you to do. Don't have any running jumps. You can have sentence fragments or run-ons, just don't have too many of them. You can have typographical errors, but once again, just don't have too many of them. You must be logical. Your report follows a standard format, and you have to use terminology appropriately. You can use it inappropriately, but you can only use it inappropriately so many times. You use it more than that, then you don't get a check in that line, and that is a failed assignment. So you know you have clear expectations as to what is expected in this thing. It can suffer from all kinds of things. But for me, grading it, I have to ask, is that so large that it makes me put it in the fail category versus put it in the pass category? And the way specifications grading works is that you have to pass a certain percentage of different types of assignments. So you can even fail some assignments and still make an A because you've reached the criteria for doing whatever is necessary for an A. You've passed. So for example, for an A, you've had to pass every one of your literature summaries. You couldn't fail any of them. For a B, you could fail one of them. For a C, you could fail two of them. If you failed three or more, you were down in the DF category. So when I shifted to doing this, I had to think about, so specifications grading is really good at testing what you can do. Specifications grading is not particularly good at testing what you know. But that's also an important thing to do. So we still had tests, but because of specifications grading, I kind of changed the structure of the test such that the test was focused on what you know, not what you could do with that knowledge. And so one of the shifts that I made is I shifted from using multiple choice tests to multiple choice tests because I'm interested in knowing what you know rather than how you can apply that. I'm thinking about how you can apply that in your literature summaries and in your lab reports and things like that. That's where I assess those things, not necessarily on a test. So what I did when I moved to specifications grading is I really tried to focus on writing exams that test what you know rather than test how you can apply that knowledge to, for example, novel situations and things like this. So yes, it's a multiple choice test because I'm really trying to just get at what you know, not more intricate levels of knowledge. And I'm going to talk about levels of knowledge here in a few minutes so that you understand this. On your literature summary, when I ask you to do critical thinking, this prospective thinking into the future, I want that to be really good. Because you're juniors and seniors, you should be capable of doing that now, which you maybe were not as good at when you were first year students. And we'll talk about kind of what that is today. So yes, I will adhere to my past rule of trying to design tests in animal behavior that really are about knowledge gain, not about application of knowledge. Does that make sense? Okay. All right. Good observation there. Okay. Other questions? Okay. All right. Good. Okay. Day one. What is the speed of light in a vacuum? What's that?
I can tell you already.
Oh, you can tell because you already pulled it up. Well, that's not the point of this.
I didn't realize.
Okay. All right. Speed of light in a vacuum, 3.8 times 10 to the minus 8 meters per second. Avogadro's number, you know this. So take it off your screens. What is Avogadro's number? Not 10 to the negative 4, this is a big number. 10 to the 23rd. 6.022 times 10 to the 23rd. What's the atomic mass of hydrogen? 1.008. Carbon? 12-ish, 12.011. Nitrogen? It's going to be around 14. Oxygen? It's going to be around 16. What would you expect hydrogen to be, though? Huh? Well, what would you? I don't expect hydrogen to be 1. That seems odd to me, because what do atoms have? What do all atoms have? Protons, neutrons, and electrons. Which of these are big subatomic particles? Neutrons and protons. And electrons are teensy weensy. So if hydrogen has 1 proton and 1 neutron, why is its atomic mass not closer to 2? When we think of atomic mass, what is the atomic mass? If you were to tell your mother, explain to her how we arrive at the atomic mass for a particular element. This is what the atomic mass is. It's the weighted average of all of the different isotopes of that element. So if the actual atomic mass is 1.007, what does that tell you about hydrogen out in nature? So there's isotopes out there that only have 1 what? Do they have 1 proton? Do they have 1 neutron? They have 1 proton. Most hydrogen out there is missing the neutron. Most hydrogen in nature exists as protons floating around with an electron attached. You knew the mass of hydrogen. You just knew it. Why? Huh? You've taken a lot of chemistry. And in chemistry lab, you had to figure out, for example, the molar concentration of something. And in order to do that, you had to know the masses of the different elements in each of those compounds so you can know, for example, the gram molecular weight of a thing so that you could do a 1 molar or a 2 molar or a 3 molar solution. And over time, carbon and hydrogen, nitrogen, oxygen, those are so common that you ended up just learning the masses because you needed them to do something, to do a particular thing. And so it just became something that you got to know over time. But what you clearly hadn't done is ask yourself, well, why is hydrogen 1.007 rather than 2? This is the difference between knowing something and understanding it. When I think about carbon, 12.011, I know 12.011 because sometimes back in my undergraduate career, I learned it the same way you learned it because I had to constantly use it in my chemistry classes. But it didn't really make the same amount of sense to me until I learned about nutrient cycling, and I discovered that carbon comes in carbon 12, carbon 13, and carbon 14 isotopes. Carbon 12 is the most abundant isotope. These are both relatively rare isotopes, which is why they bump up the average weight a little bit rather than a lot. Because most carbon is carbon 12, a little bit is carbon 13, and a little bit is carbon 14. In C3, C4 photosynthesis, though, when we think about that, carbon 12 is much easier for plants to fix because it weighs less. That little tiny difference in weight actually matters at the molecular level in terms of organisms fixing carbon. Carbon 13 and 14, because they weigh more, actually are less often used in photosynthesis and incorporated into sugar, unless you're a C4 plant. Talked about C3 and C4 photosynthesis in plant bio. C4 photosynthesis, the stomata stay closed for longer periods of time, which means that the C4 plants leave their stomata closed until they have exhausted all of the carbon that is in the carbon dioxide that is in the leaf. And only after they've used all of it up do they open their stomata, let more carbon dioxide in, and then they close their stomata back again. And then they proceed to use all that carbon dioxide that is up in there because they can, because they have C4 photosynthesis. They use PEP carboxylase instead of rubisco for initial carbon capture. And what this means is that if you look at the ratio of isotopes of carbon in C3 and C4 photosynthetic plants, C3 plants have almost exclusively carbon 13 in their bodies. C4 plants include a higher proportion of carbon 13 and carbon 14. So when you look at the carbon that is in C4 plants, you can see that they have a different isotopic signature than a C3 plant because it uses up all of the carbon that's easy to fix and the carbon that is heavier and as a result is harder to fix. Then all of a sudden, not only do I know what the mass is, but I know that the mass is related to isotopes, and I understand how that enables us to look at the bodies of organisms. We could take your fingernails and your hair, we could do an isotopic analysis of them, and we could determine how much corn you eat. Because corn is a C4 plant, not a C3 plant. Now I have all this understanding about that number that I didn't have before. Different scales of knowledge, different levels of knowledge. Knowing and remembering factoids is one level of knowledge. Understanding all that I just went through, that's another level of knowledge. That's still knowledge, but that's knowledge that is more than just knowing a thing. It's knowing a thing, understanding that thing, applying that thing to what you guys are eating and drinking. Do you understand that distinction? As juniors and seniors, we want you to be doing the understanding and the application rather than just the knowing. Those are the kinds of things I'm going to be expecting of you in this class. And I would be expecting that of you whether I was grading this with specifications grading or some other form of grading. So let's think about this in real terms. You guys have seen pictures like this in the past. What's going on here? May have to reach back 3 years or so. We have horned lizards, and what has happened to these horned lizards? They're dead. Well, who's killed them? A bird, a particular type of bird. Do you remember which kind of bird? A shrike. Yep, shrikes. Shrikes kill their birds how? By what? By impaling them on sharp objects in their environment. Shrikes do this for a variety of reasons, potentially. And there have been a number of hypotheses that have been put forth as to this particular weird behavior of loggerhead shrikes. What are a few of the hypotheses that would explain this behavior? So here's a shrike. It has impaled another bird, which is really crazy. The bird is not much smaller than the bird that did the killing. What are some explanations for why shrikes might do this? Okay, so one thing that could be happening is they have really weak feet. They have kind of a hawk-like beak, but they're not a hawk. They're not a raptor. They're a passerine bird. And as a result, they don't have heavy talons like a hawk or a falcon does, or an owl does. And so this might just be an efficient way of killing an animal that is close to its own weight that it can't accomplish with its beak and its talons. So that might be one hypothesis. What's another hypothesis? Because it has weak feet, what can it not do with the carcass of that prey item once it's killed it? Can't grab a hold of it very easily. Hold it in place while it strips the food from it. So it might also be a prey handling issue. Why else might you stick prey on these barbs all over your territory? Might show other birds that this is your space. What else might you be doing with these prey items? You might be showing off to a mate. Very good. What else might you be doing with these prey items? Another hypothesis might be that you're storing these prey items for future use. When you look at impaling behavior of shrikes in the literature, in textbooks on animal behavior, et cetera, generally you are going to find two reasons proposed for why shrikes do this. One reason is it helps them handle the prey. It enables them to secure the prey so that they can strip the flesh off the prey because their feet are too weak to hold on to it themselves. Not a problem that falcons and eagles and hawks and owls have because they have big, strong talons. The other reason that is put forth in textbooks is that this is food storage. It's forming a cache, food caching. They're storing this food for later use. But look at this frog. That frog is dried up. It's dried up in an L shape. There is no way that that bird is going to come back and be able to eat that frog. It is too misshapen in a shape that can't allow it to swallow the animal whole. It's all dry and crusty. You can't strip the flesh off of that even though it is impaled. That bird with that frog is not storing that frog for later use. When I was in grad school, I went out into the field collecting spiders, and I saw a barbed wire fence at my field site. And on this barbed wire fence, it was spring, so frogs were out breeding. And on this barbed wire fence were a bunch of frogs, and they were all impaled on the barbed wire fence. Their heads were here, their front legs were here, and their back legs were hanging down like this. And they were dry and crusty. Tons of them on this fence. And I thought to myself, what's the explanation for why shrikes impale prey? So I saw this and I knew that was done by a shrike. That's impaling behavior. I also knew impaling behavior has been described for food caching and for food handling. But when I looked at that fence, I knew it couldn't be either of those explanations in this particular case because those aren't the only two hypotheses. Another hypothesis is maybe you're using this as a display to advertise to females what a good hunter you are. Why would females care how good of a hunter you are? I want to hear from Avery. I've heard nothing but you. No, it's fine. Speak up. Okay, so if you're choosing a male to mate with, you're mating with that male to have babies. If they have advertised that they can provide well for your offspring, better to mate with a male who has a large cache of prey items in his territory than a male that has a smaller cache or no prey items in its territory. That would indicate that either that male is a better hunter or that male has a better territory that just has more food in it, so then hunting is more efficient. Whatever it is, from the perspective of the female, like that's a good quality male because he has lots of food available to him. He has so much food available to him, actually, he has food he's not even getting around to eating. I went back to my major professor at the University of Texas at Arlington, and I said, I don't want to work on foraging behavior of spiders anymore. He's like, what do you want to work on? I said, I think shrikes impale their prey as a sexual signal to females to advertise male quality. Explained to him what I saw out in the field. He's like, that sounds really cool. What do you think he told me to do next? No, before that. Whenever you have a new idea, what's the first thing you should do? You should get to where? It's close to home. It's right across campus. Go to the library and do what? Yeah, find out if anybody else has had this idea. You're a first-year graduate student in your PhD program. Surely you're not the first person to have thought about this. Guess what? It's 1991. I was not the first person to have thought about this because Joseph and Pinchot published a paper on it in 1989. Which is the paper that I want you to read for Friday. Knowledge is one thing. Knowing those frogs were put there by shrikes. That's knowing. Knowing that shrikes can do this for prey handling and for prey storage. Knowing. Seeing the frogs and understanding that's not what these things are doing there because they can't really be eating that. And then saying, okay, well, if it's not prey storage and it's not prey handling, what could it be? Requires then going and finding other knowledge that is rattling around in my brain because I've had a course in animal behavior at that point in my career. I know that sexual displays are a thing. I know that bird territories are a thing. Knowing that there's maybe another hypothesis, figuring out that there's maybe another hypothesis out there that I had never been told about because it doesn't appear in any of the books, because it wasn't even published until 2 years before I saw this. That's understanding, applying, thinking forward into the future, thinking about things that people haven't thought about before. So we can think about facts, atomic mass of things, talk about facts about behavior of shrikes. We can also think about the conceptual underpinnings and theories about different areas. But then there's also this way of thinking that synthesizes information, takes information that doesn't seem to go together and says, okay, these frogs don't go with those hypotheses. What's another hypothesis out there that might explain this? That's synthesizing information and creating something new. So have you guys ever heard of Bloom's taxonomy? It's a hierarchy of thinking in a sense. The first thing you have to do is you have to know and remember stuff. 12.011, atomic mass of carbon. But then you have to understand that. Know what's behind that number. What does that number really mean? Applying that to something like C3 and C4 photosynthesis. A higher level of thinking beyond just knowing the thing and just understanding the thing. Now you're applying it to something else that you might find interesting, which is like the photosynthetic systems of plants, perhaps. I mean, I don't find that interesting, but other people do. Analysis and synthesis and creativity are the top levels of thinking, and they build on one another. To know something, you need to know something before you can understand it. You need to understand it before you can apply it to anything. You have to be able to think about applying it to things before you can do any sort of analysis and evaluation. And ultimately, you need all of these lower levels in order to create something new. And if you're a biologist out in the world, whether you're a medical doctor or a researcher or whatever, you have to be thinking at those higher levels. A person comes in to your doctor's office and presents a set of symptoms. You have to rely on all of the things that you know and remember, and you have to analyze them and evaluate them to ultimately figure out what is actually wrong with that person. I got bit by a cat last semester. Working in a barn, stepped on a cat because the cat was underneath my feet all day and I stepped on its tail. It bit me in the leg. A couple of days later, my leg starts getting gray up and down the leg. It's obviously gone septic. I went to the doctor and showed him the bite. And he's like, "You got bit by a cat, didn't you?" I'm like, "Yes, I did." He's like, "That looks exactly like a cat bite looks." Because he saw the wounds, he saw the sepsis, it was down at a part of the body where you might get bit by a cat if you stepped on their tail. He took a bunch of disparate pieces of information and put them together into a synthetic idea where he knew what had happened to me before I even told him what happened to me. Plus, cat bites are well known for going septic. It's that level of thinking that we're going for. So be working towards that as you go through this class. going to be asking you not to just know things, but I'm going to want you to understand those things, be able to apply those things to novel situations. And at least in some parts of the class, in the literature reviews, in the literature summaries, I want you to be projecting and creating into the future. So that's kind of the goal. Questions? All right, I'll see you guys tomorrow at 10:45 then. In lab, not anywhere else. There are some video clips that are links in that PowerPoint. Go and look at those. I skipped over them in class because of time, but go and look at them. They're informative, but not absolutely necessary.